I just want to tell you something that's fun. And then I'm going to introduce our great speaker, David Fitzpatrick. You know, the Institute, the reason we started this Institute over 25 years ago was to bring people together to talk about ideas. And we never knew what people were coming or what people weren't coming. But we've been doing it for over 25 years. Can I tell the table? Well, I have two scientists here. One is a genius in music who's just finished writing a book about the brain and music. And then we have David Fitzpatrick. And all of a sudden, we are having a hell of a good dialogue between the two of them. They have never met, and maybe would never have met. And next year, I'm going to see if I can't get them together to have a dialogue together to see what they come from. Because, you know, we do music at the Institute, and the only reason we can do the great level of music is because my friend Ford is one of the greatest teachers at Curtis, and he has these stars. A lot of them are very much prodigies like he was. So all I can say, I've been so fortunate. And of course, he has a wife who was the Metropolitan Opera singer. So that is why we can have some fabulous music. Okay, now I'm going to introduce David Fitzpatrick. He is the founding scientific director and CEO of the Max Planck Florida Institute. Prior to this, he served as the James B. Duke Professor of Neurobiology at Duke University of Medicine. Wait a minute, and here it goes. He is an author of over 90 scholarly papers. Dr. Fitzpatrick's scientific contributions has earned him international recognition in the neuroscience. Today, he is going to present on the topic of curiosity-driven discovery, illuminating our path to the future. I think that's a heck of a topic. Come on up.
And Dale, thank you so much for inviting me here today. We actually met by accident in Aspen, Colorado. And I understand a number of you have been in Aspen, and it's a wonderful place. But we just started talking, and she said, "Why don't you come and do this?" And I said, "Wow, I'd love to." So thank you all for being here today. Curiosity-driven discovery. This is something that I am passionate about, and I think it is critical for our future. And I hope by the end of this talk, you have a sense for what curiosity-driven discovery is and why it is really critical that we support it. Our institute, the Max Planck Florida Institute for Neuroscience, focuses on the brain. And I don't have to say it, but I will. The most complex living structure in the known universe. Billions of neurons, trillions of synapses, astounding capabilities. I'm sorry, I feel like you can't see. I'm right in here. So, actually, maybe I should go over here. How's this? Is that a little bit? Oh, sorry. Okay, I'll just keep banging back and forth. So, the brain, it is truly amazing. Sensation, movement, learning, memory, language, cognition, all of these things are due to our brain. And without doubt, it is, in a real sense, we are our brains. And the brain does such a good job of creating us that, we don't even recognize it. We take it for granted. And, if you just think for a moment, I'm standing up here. My brain is formulating thoughts. Those thoughts are turning into words. Those words are turning into muscular contractions that ultimately lead to sound coming out of my mouth, coming into your ears, and turning into a thought. I'm sorry, that is amazing. That's the neural circuits in your brain that is making that happen. Now, as I said, we often take it for granted. And when I was teaching back at Duke, I taught the medical students, basic neuroscience and whatnot. I would try to get them to understand how amazing the structure is in a really simple way. Okay, here's a simple way. You know, when you look around the room and you see light and dark, oh, come on. That's not much of a calculation for the brain, right? That's just in the world. Okay, light and dark there, right? Watch what happens when I cover up the border between those two. Okay, so now, would you have thought that these were actually the same contrast? No, of course not. But our brains are doing a computation on that image to give you that sensation. Here's another one. And I don't know if this is going to work in the lighting here, but when you look at this, does it seem to be moving? Do you see motion? You think this is a video, right? There is no motion here at all. Your brain is constructing the motion. Okay, now so your brain constructs motion. These are all very simple things, okay? But your brain is doing a computation to give you that perception. Can we turn the light down just a little? Is that possible? Or is it, I guess it's down. I guess it's just the light from outside. What do you see when you look at this? Black and white castle. Would you believe that your brain can turn that into a colored image? Thank you for saying that. Okay, I want you all to look at this spot here just for a minute or two. Just look at that spot. It's really boring, I know, it's just a spot. Okay. Did I get you with that one? Okay, okay. So again, this is the neural circuits in your brain that are giving rise to your perception of color in an image that has no color. So that is certainly one way to give you a sense of the tremendous job that your brain does. This is another way to consider all of the disorders of the brain that impact our lives. And I am sure that everyone in this room has a family member or a friend who has been impacted by one of these disorders. Did you know there are 1,000 disorders, neurological psychiatric disorders? There are over 700 million people in the world who are impacted by neurological and psychiatric disorders. And yes, directly. And 1.5 trillion is the amount of dollars in the US annually that are associated with treatment for these disorders. If this is not a huge problem, I'm not sure what is, okay? Now, how can we make progress here? Okay, we need progress. How do we make it? This is one of my favorite scientists. This is Max Planck. I'm sure you've heard of Max Planck over the years, an amazing physicist. This is the most famous quotation from Max Planck. Insight must precede application. Now, sometimes when I say that, I get these looks like, what does that mean? So this guy has another version of it. You can't fix it if you don't know how it works, okay? That's really what we need. We need to understand how the brain works so we can fix it. Now, the question is, how do we get these insights? This is where we get to curiosity-driven discovery. Now, whoops, sorry. What is curiosity-driven discovery? Well, it's allowing scientists the freedom to pursue novel lines of inquiry, to uncover fundamental principles of nature we didn't know and we couldn't have imagined. Okay? It is really going beyond what we currently know. And I think I want to make this clear in the beginning, it's critically important to take everything we know and push it as far as we can. But we can't assume that we know it all. And in terms of the brain, there's so much that we do not know. I call curiosity-driven discovery the engine of innovation and the foundation for all applied research and technology. Now, in trying to get people to understand the impact of curiosity-driven discovery, lots of times I'll start talking about, how these discoveries in physics, have led to our cell phones, have led to X-ray technology, and so on and so forth. This is a book that just came out recently that actually is called by Steve Kaplan, who's at University of Nebraska. But it does a really nice job of breaking down how all of these things that you see here that are critical for medicine, all of these things were derived from curiosity-driven discovery. And I've got a couple of here, CRISPR-Cas9. I'm sure many in the room have heard of CRISPR-Cas9. Fundamental basic science. This is changing how we can alter gene structure, okay? mRNA vaccines. In COVID time, that was amazing how quickly the vaccine was created. How did that happen? Because we had people that had explored mRNA and understood how it worked and how it could be used to create vaccines. Okay, 90% of Nobel laureates are curiosity-driven researchers, and some have worked on their questions for over 20 years. I'm not a Nobel laureate, but I've worked for more than 20 years on things. Okay. Now, it's always nice to go and see what Albert thinks about these things. So this is a quote from Albert Einstein. "I have no special talent. I'm only passionately curious." I might argue with that. I think there was some talent there, okay. But, okay, the most important thing is not to stop questioning. Curiosity has its own reason for existing. Now, here's another Nobel laureate, Aaron Klug, got his Nobel Prize in chemistry. And I think he also says it well. Human curiosity, the urge to know, is a powerful force and perhaps the best secret weapon of all in the struggle to unravel the workings of the natural world. Now, this leads me to the Moxbox Society. And I like to think of the Moxbox Society as the home of curiosity-driven discovery. The Moxbox Society, its home is in Germany. There are 86 institutes. There's only one outside Europe. That is the Max Planck, Florida Institute for Neuroscience. But these institutes cover the full range of science. There have been 22 Nobel Prizes awarded to members of the Max Planck Society. And this is me commenting here. If you're a member of the Moxbox Society, it's a scientist dream come true. Seriously, it is really an amazing opportunity in your life to pursue science in a way that you haven't had before. Moxbox Society is the #1 nonprofit research institution in the world, as judged by Nature Index. So, what is it that creates an environment for curiosity-driven discovery? So, it's investing in talented individuals, providing the environment in which their passion, curiosity, determination, and intellect can be realized. Guaranteed annual research support for the scientist. What does that do? It lets you take chances. High risk, high reward. Access to the latest technology and equipment. I hope some of you will visit us up in Jupiter. You will be blown away with the technologies that we have within the Institute. Freedom. The scientist has the freedom to explore what they are passionate about. Also to take the risks that yield the rewards. And, you know, as a scientist, you gotta know what you know, and you gotta know what you don't know. That's why it's really important to have people around you with the technical skills to enable the science that you really want to do. And we have that up in Jupiter. So just a moment here about the Institute in Jupiter, and then I'm going to talk about what we're actually doing. So this, how did we get curiosity-driven discovery in Florida? Shared vision, I'm sure most of you know the person on the left there, Jeb Bush. And that is Peter Gruss on the right. Peter Gruss was the head, the president of the Max Planck Society. They put their heads together and said, yeah, let's have a Max Planck in Florida. And they made it happen. And they deserve an enormous credit for making it happen. That was 2007. We had a Nobel laureate come over early on, 2009, Bert Sackman, to get it going. I signed on 2011. 2012, our building opened. I recruited another scientific director, Ryohei Yasuda. And 2018, you see the fireworks over the Institute there? Do you know what that is? We were brought here by resources from the state of Florida and Palm Beach County. The fireworks are because Germany recognized what we had achieved and provided us with sustainable funding for the future, okay? Thank you. Yes, yes. Yes, absolutely, absolutely. So, what are we doing? We're focused on neural circuits, structure, function, and development. Development and application of new technologies for probing the structure, function, development of neural circuits. We use experimental approaches that bridge different scales of analysis, from synapses to networks, and integrate molecular, physiological, anatomical, behavioral, and computational approaches. I know that was a mouthful, okay? But I'll break it down. What are neural circuits? We can start there. So let's just start with the part of the brain that is really critical for almost everything that's going on in this room right now. You know, seeing, hearing, feeling, moving, cerebral cortex. This is the outer portion of the brain. Gray matter, you've all heard of gray matter. white matter, a little deeper in the cortex. The gray matter is where all the neurons are. They're doing the computation. The white matter, that's the cables that are supplying information to the cortex and bringing it away. Now, the cerebral cortex, this outer portion here, is a folded sheet of neurons. And it's hard for people to understand the size of this thing. So you look at the width of a nickel, okay? That's the width of your cerebral cortex. That's the width of this part of the brain that is giving rise to your conscious perception, okay? Now, let's take a look at what's in that nickel. And you have an extremely complex neural circuit with all of these neurons sitting there, beautifully colored here, not colored in your brain, but beautifully colored here so you can appreciate the different types of neurons there are. Now, let's take this a little further. I'm starting, what is a neuron here? What is it actually doing? And I'm sorry if this is going to sound like I'm teaching. Okay, I can't help myself. This happens. This is a neuron, a single neuron. This is a single cell in your brain that is giving rise to all that we are perceiving here. Now, the neuron has a cell body. That's this little black guy down here. And it has all these processes that come out. Those processes are called dendrites. The dendrites receive information. And the axon sends information, okay? So incoming information, information leaving. And then there's synapses. And the synapses transmit the information between neurons. And if we blow up a little piece of it here, this is the dendrite. This is a piece of the dendrite. It's called a dendritic spine. Here's the axon from another neuron sitting right next to this dendritic spine. And this, the junction, is the synapse. So, how does this work? Okay, electrical activity, release of neurotransmitter, a chemical, electrical activity on the backside. And that's really the basis for neuronal function, okay? So we have a neuron sitting here. Lots of the synapses on that neuron are getting activated. And sometimes enough of those synapses get activated and the neuron fires and sends the information to other neurons, okay? That's the basis for neural function. Now, let's talk about the reality of things here. There are 20 billion neurons in your cerebral cortex, 125 trillion synapses. There are about 5,000, 12,000 synapses per neuron. There's a diversity of cell types in your cortex. This leads to all sorts of problems answering these questions. What is the wiring diagram of the cortex? We don't really know yet. What is the neural code, the pattern of activity that gives rise to brain function? We don't know. How does experience change circuit activity? We don't know yet. And this question, how does disease alter circuit function? We ultimately want to know that, but we have to answer those other questions so that we can really go after the diseases and understand how they impact this circuit. Okay? You need to understand how it works before you can fix it. So, in the Institute, we are unlocking the brain's mysteries, and I'm going to tell you about some technologies that a number of the investigators in the Institute are using. And I'm telling you, these technologies, if you had told me we could do this 5 years ago, I would have said, yeah, sure, okay. These are really amazing advancements. Seeing circuits in action, I say, and manipulating circuit function. What's the common element here? We're doing it with light, okay? So hold on to that thought. So, when I talk about seeing circuits in action, what goes into your head? I bet most of you think, oh yeah, functional MRI. That's how we see circuits in action, right? Sure. Spatial resolution of functional MRI on the order of centimeter. You've got so many neurons in that centimeter, and you're actually looking at a vascular signal. So you don't really know what's happening with different types of neurons with fMRI. This guy, Winfried Denk, Max Planck Institute, Martinsried, Germany, he was curious. Can I use light to visualize the activity of individual neurons in the living brain? And he came up with a way to do this. This is 2 photon in vivo imaging. I'm not going to take you into the physics of how this works, but I will tell you, and if you look at this image here, this is what happens if you project light into a fluorescent liquid from a normal microscope. This is what happens when you do it with 2 photon imaging. You get a point. What does that allow you to do? That allows you to focus in fine detail into the brain. Now, that gets us an image. That doesn't get us activity. How do we see the activity of these neurons? Well, again, it's a trail of curiosity. And believe it or not, it began with understanding why jellyfish glow, okay? By trying to figure out what it was that caused jellyfish to glow, and actually getting a Nobel Prize for it, we established green fluorescent protein. And this is genetically encoded. And then we had another curiosity-driven scientist, Lauren Luger from HHMI, who figured out a way to make this a calcium sensor. So this molecule, when it sees calcium, beep, beep, it gives off light, okay? Now again, there's a lot of light in this room, so it may be hard for you to see, but hopefully you're seeing some little dots light up here. You see that? You're looking inside the brain. This happens to be one of the creatures that we use. This is a tree shrew, and if you saw it, you would think it was more like a rat or something like that. It's a very small animal, but it has a beautifully organized cortex. But this technique, 2 photon imaging, allows us literally to see the activity of neurons in the living brain while this animal is looking at a video screen. And we are able to use this technology to explore a variety of different things, okay? Seeing, eating, walking, decision-making, learning, memory, emotion. All of these things are being explored at the Institute. And almost all of them are using this technology. Now, I'm curious. I shouldn't have said that, I'm sorry. Okay, I'm curious. Have you been noticing anything about the patterns over here? They're changing, that's right. This is what the animal is looking at. And as we change, this is visual cortex, and as we change the orientation of the edges, the pattern of activity in the brain changes. So we can actually visualize how individual neurons are responding. And again, we can color code it. And so we know which neurons are active for which orientation. That is a remarkable step in the right direction. Taking it even further, we have ways of defining different cell types, different neurons in that field of view, based on their genes. What you're looking at here are different types of inhibitory neurons. So we can say what the neurons are, and we can say what their activity is. Okay? So you might think, okay, great, got the neurons. I'm going to tell you, next, we went to the level of single synapses in the living brain, okay? This is huge. To literally being able to pick up a portion of a dendrite, present stimuli, and see each one of these little things lighting up here. That's a dendritic spine. That's a synapse being activated. What does this let us do? And also, just to, sorry to give you a perspective, the human hair, did you realize the human hair had such a range of diameters? I mean, that's, I never realized that. But this is showing you it's 17 to 180 microns. You can see that these are in the order of, 2 microns, something like that. These are really tiny, but you're seeing it in the living brain. What does this let us do? It lets us take a neuron, define the inputs to that neuron through its dendrites. So we get the input coming in. And we can then compare it with what the cell body is saying. Okay, I hope you understand what that means. That is cracking the neural code. We're really understanding how neurons are taking their inputs and turning it into an output. Now, we started with cells, came to synapses. Now, I'll take you inside the synapse. This is my colleague, Ryohei Yasuda. He is an amazing scientist who has found all sorts of amazing ways to label the molecules within a synapse. Okay? And what can he do then? This is a synapse here, and you're going to see it undergo change. You're going to see it get bigger, and you're going to see it get red. Do you know what that is? That's a particular molecule that is being expressed in that synapse. It's called CAMK2. I won't go into what it is, but it's a molecule that we now know is critical for synaptic plasticity, for strengthening of synapses. And Ryohei is looking at many different molecules within the synapse and understanding how they work together to build neural plasticity. All right, the last thing I want to leave you with is manipulating circuit function. We've been talking about imaging it, seeing it. But did you ever think you could use light to change the activity in the brain? And I'm not talking about putting light on your retinas. I'm talking about actually stimulating neurons with light. And again, it's another trail of curiosity. Someone was curious about how algae are sensitive to light. How did you go from algae to the brain? Isn't this crazy? Okay. They figured out the protein in the algae that made the algae sensitive to light. Took the gene for the protein and expressed it in neurons. So now you can cause neurons to fire just by flashing blue light. Okay? This is huge. Right? This shows you're looking down on the surface of the brain. There's some cell body targets that have been identified. And over here, what you're seeing is light being shown on those individual neurons. And you're seeing them light up. And that light up is the calcium signal that tells you the neuron's active. Okay? So, we can read and write patterns of activity in neural circuits. So we are using this to explore all of these brain functions. A lot of the work is done in mice by literally stimulating the neurons in the brain. But I want to show you something that we're also doing, another species. So believe it or not, biologists still use the fly, Drosophila, because we have a wonderful understanding of the genes, and now we're getting to the point where we have an understanding of the circuitry in the fly brain. This is from Salil Bidai, who is a research group leader in the Institute. You'll notice every once in a while, a light will show up here. And we have optogenetic probes in different types of neurons in the fly brain. On this side, watch this fly when the light comes on. See what it does? It goes backwards, okay? That's because, and in fact, he gets credit for discovering moonwalker neurons, the neurons in the fly brain that, when active, cause the fly to move backwards. Here's another, bolt neurons. That light comes on, that fly takes off, okay? So you understand, we are controlling the activity of the neurons in the fly brain and generating this consistent behavior. So we're understanding the neural circuits that make locomotion and the direction of the locomotion possible. These are the scientists that have labs at the Institute, the two directors and a number of research group leaders. And all of those topics that I listed there multiple times, each lab has its own topic that they're really interested in. I encourage you to go to our website and see what they're doing. Just to give you a sense of where we've been since 2012, and okay, I'll brag here a little bit, why Max Planck was comfortable supporting us. We delivered. We delivered in terms of the number of publications, high quality of the publications. Those of you in the room who know something about science, if you get a Nature paper, a Science paper, a Cell paper, you're a happy person, okay? These are exceptional journals. A grant support, 68 mil, that's not too bad. We had 2 NIH Pioneer Awards. Very few of these are given out across the entire US. The next one, all my years at Duke and all my years here, I've never seen a person get a perfect score on a grant application. It wasn't me, okay? One of them was a research group leader. One of them was my fellow director, Ryo Yasuda. A perfect score. That means the review panel of other scientists gave it, absolute, this is it, no need to talk about it. Our success rate for grants, 36%. And our progress, here's another number, 255. That's the number of trainees that we have had in the Institute. Thank you, thank you. 255. We are so proud of what we are doing to make sure that we have a next generation of curiosity-driven neuroscience. And to be honest, we show you all these great things, the fact that we're able to do all this stuff. Do you know who's doing this? Trainees, the postdocs, the graduate students, the postdocs. Yes, they're being guided, but they're the ones at the bench making that happen. So we have, this is all the different levels we have. We have a high school program that is absolutely wonderful, and Laria Drago, who is sitting at the table back there, is responsible for this program. This is an amazing program. Every summer we have high school students. I cannot believe they give a presentation at the end of their internship, and I'm like, oh my God, could I have imagined doing something like that when I was in high school? Like explaining to me how super resolution microscopy works. No way, no way. We also have undergraduate students from FAU, we have postbac fellows, we have graduate students, and we have postdoctoral fellows. And we've called this the Max Planck Academy. And I'd like to play for you a little video that gives you a sense of the philosophy that surrounds the Max Planck Academy.
Close your mouth, all right.
Becoming a scientist is more than a matter of special training. It starts with curiosity, fueled by a sense of wonder, and a motivation to discover the unknown. It's learning to ask the aspirational question: and being given the chance to take the risk to step forward and see what happens. Believing that a different perspective can open up new possibilities. Our philosophy at the Max Planck Academy is to provide the much-needed support that is necessary to pursue a career in science. We inspire scientists at all levels of their career, supporting their fascination with how the world works, turning this sense of wonder into novel insights that redefine the field of neuroscience, where researchers are given the freedom to work with technology that is unavailable anywhere else. It's a place where a high school student can discuss big ideas with Nobel laureates, where college students can see networks of newly understood neurons years before they're described in school textbooks. Max Planck Academy. No limits, just science.
Okay, now I'd like just to end with this. I hope I've given you a sense of what we've accomplished up in Jupiter, and very proud of what we have done there. Wondering if you might be curious about the only Max Planck Institute in the US. Curious about it? Let that curiosity drive you to Jupiter for a visit. We would love to have you visit. If you are interested in visiting with us, please talk to one of the team, the development team, who are here today. And I'm sure they would be able to find a time to have you visit and see firsthand the amazing science that's happening there. And thank you all.
When we met, you told me that you were able to go in deeper than you've ever been able to go into the human brain. A live person, right? Not human? Okay, I didn't understand. So you have all this research that you're doing. Who's taking this research and make it actual for us as a human being?
I mean, I think that's a great question, and what you're asking me is, how do these fundamental basic science discoveries get translated into something that is a drug, is a pharmaceutical, is a diagnostic, et cetera. Or on the operating side. And you have to appreciate how long it takes to go from this discovery to something that is being delivered therapeutically. That takes a long time. But what I will tell you is, if you're looking for the shortest version of this, I would argue, would be trying to define the molecular pathway that's involved in memory formation. I showed you the synapse. I showed you all of those molecules that are interacting. I didn't explain to you how you can look at the molecules when they're activated, who they're interacting with, and how that leads to strengthening of a synapse. When we know those molecular pathways, as you can imagine, every one of them has potential for therapy, either blocking it or doing something that increases it, right? So, there's nothing in the Institute that is ready to go out the door as a therapy. There is not. But what my argument would be is, this isn't fair. I mean, we all know the tremendous challenges that Alzheimer's has been. And the number of times that we hear, here it is, and then it isn't, okay? And so, as I said earlier, I think what we need to be doing is taking everything we know and trying as hard as we can to do that. But what we're doing is trying to say, wait a minute, there are other things here that we never imagined, that we didn't know, that could end up being something that is relevant for Alzheimer's. I don't know if I've answered your question, but okay. This would be a biotech. So biotech companies would take this discovery. Now, one of the things that's always a little bit tricky here is we publish this work. That means it's out there and it's available, and then it can be turned into something for a profit. We are nonprofit. That is not what we do. But what I would say is, we do have some inventions. So, for example, Ryohei Yasuda has come up with a form of imaging called FLIM imaging, fluorescence lifetime imaging. And that is something that is in the patent stage and so on and so forth. So that potentially could be something that would bring revenue to the Institute. But that is not our goal, okay? The goal here is not to make money. The goal is to understand, okay? That's the goal. Yes?
So you touched on Alzheimer's, and that's what I'm curious about. The light therapy, are you using that as a thought to possibly effectively change the molecular structure of the brain?
Well, go ahead, I'm sorry.
Spinning off and getting those tangles in the brain. Have you studied that, and does light therapy show any?
So you understand that in the examples I'm showing you, we are expressing proteins in those neurons in order to get them to respond to light. And as it turns out, we're doing that with viruses. So we literally use a virus to express, and this virus we're using, trust me, is very safe. It's actually a virus. It's actually a virus that is used. It's called AAV. It's a virus that people are comfortable seeing it be used in humans, which is really cool, because if we could get this to the stage where we could use it in humans to stimulate neurons, that could be huge. We are nowhere close to that. I want to make sure you understand. I'm not promising that. But that's certainly an interesting possibility.
Hi, thank you. My son, who was born with a neonatal stroke at birth, has cerebral palsy. So how can your treatments come to light to help some individual like himself? And how soon can those treatments come to light?
Yeah, again, you've used the word treatment. And what we're doing here is discovery, not treatment. But what you just mentioned is really an important element of what we do. A good part of my lab is focused on development of the brain. You know, all those circuits that I just showed you, you have to understand those circuits are really immature at birth. Most of those synapses form after birth, under the influence of experience. And we are trying very hard to understand how experience is building those circuits. And I don't have the time to go into it, but if you're interested, I can tell you more about that. It's remarkable how much experience plays a role in building those circuits.
Yeah, a jellyfish, which you show, doesn't have a lot of parts. But when it glows, is it thinking?
That was very cute. And I'll give you a cute response. I don't know. That was very cute.
Brains function via neural circuits, and computers function via neural circuits. What do you think of do you think there is a possibility that someday a brain-machine interface could be developed?
Absolutely. Yes, yes. I mean, I think that that's, how do I say it? What we're trying to do. I showed you the imaging side. We're also doing things with multi-electrode arrays that allows us to record large numbers of neurons simultaneously. So I presented this imaging as the most amazing. It is. But I can imagine some of you are saying, what's the temporal resolution of that imaging? How does that relate to what neurons are actually doing? And that's why we're actually now getting set up to do voltage imaging in the brain rather than calcium. But for the moment, multi-electrode arrays are the way to have the temporal resolution. But you understand the problem there. You have the temporal resolution, but you don't know what you're listening to. You don't know what the neurons are. So when we can get up to the speed where we have the temporal resolution and the spatial resolution to know which neuron's doing what when, literally, I will be shocked if we're not in a position to selectively activate circuits by focusing on particular cell types within that circuit to get a particular effect. Yes? Yes, so I think for epilepsy, what you would like to have is a way to turn those neurons off, okay? And it went by so quickly, I'm sure you didn't get it, but what we are now able to do is to identify different subpopulations of GABAergic neurons. GABAergic neurons are the inhibitory neurons in the cortex. And so if we can figure out a way to turn them on, you know, when a seizure is detected, then this would be a way to handle that. Are we there yet? No, we're not. But yes, because you're talking about you know, activity patterns and trying to change those patterns. Yes.
So 2 questions. First, I thought there was a Max Planck Institute of Math at the New York University. Maybe it's the Fermi Institute?
There is a computational. Are you talking about a computational?
Yeah,
Yes.
Institute of Math for a long time at the New York NYU.
Well, it's not. So I said we're the only one outside That's not an institute in the sense of like an institute in Germany.
Oh, okay.
That's an affiliation with the Max Planck Society. This is a Max Planck Institute. I'll have the vice president of the biomedical section of the Max Planck Society in town in May for our board meeting, evaluating what we're doing, and so on and so forth. So it's,
Okay,
and in fact, honestly, the people are surprised when they come from Germany because this institute is just like the institutes in Germany in that regard.
That's probably right. But the second question I have is, which is similar to what was raised before. Now, I mean, you're doing a very, very basic research, but are you coordinating with other medical schools and like Harvard University schools, things like that, who are taking your research maybe to the next level? For example, what role synaptic plasticity play in various diseases? And the fact you can visualize it, now it plays a role. So are they using your visualization techniques into others, or you're very just self-centered and not coordinating with other places?
I was just in Boston last week for a big brain imaging session where we were talking about all of the new developments. And to be honest with you, I haven't presented the latest because we're sharing that just among the scientists who are doing it. So we share as a community all of the work that we are doing. And in terms of medical, try again. In terms of medical schools and being involved, of course. I mean, this is how do I say it? Anytime there's any possibility that what we have would be relevant, we share. That's what this is.
Thank you for your insight, very informative. I just wanted to ask a question. When there is contusion to the head, would you happen to know why white matter develops in the brain?
You know, it's really important when you're a scientist to be able to say, you don't know. And honestly, I don't know. Sorry, I wish I could tell you something.
You've indicated a lot of things that you don't know or that are unknown, but there are a number of practical applications that have grown out of the study, curiosity-based and otherwise. I mean, for example, the deep probing for treatment of Parkinson's.
Deep brain stimulation, yes.
Deep brain stimulation, yes. And I'm sure that you could give us several other examples.
Well, I don't know if you remember my slide, but that slide had all of these developments that are related to medicine. I mean, come on. If you have an X-ray, you have a functional MRI. All of these things originated, many of them actually, from physicists, who laid the groundwork for doing that. But you needed the biologist to say, wait a minute, I wonder if we could use that in the brain to actually visualize the brain. And then somebody had the idea, well, wait a minute, if we did this in a way that we could see changes in vascular supply, since from the biology we know that changes in vascular supply are associated with brain activity, maybe we could actually visualize activity. And so that's how we got to fMRI, okay? So, and I think as much as I, was sort of dumping on fMRI because it doesn't have the resolution, in terms of being able to see into the human brain and diagnose, that's huge. Okay, so those are some examples of things like that. You mentioned deep brain stimulation. Deep brain stimulation is remarkable. I mean, sticking electrodes in the brain, going into the regions of the brain that are relevant to Parkinson's disease and stimulating, that's huge. But what I will tell you is that's an electrode that is stimulating non-specifically all of the neurons that are sitting there. And there are side effects to that deep brain stimulation. And we wonder whether it could be done with cell type specificity, whether that would improve the effectiveness of it. And we actually have light fibers now that we can put into the brain. And so if we had the optogenetics expressed in the neurons that are relevant, and then could use light to drive them, that would be huge. Okay?
Thank you.